again, good morning from Florida, and thank you, John, for giving me the opportunity to present some of our research. And I think most of us who work in the marine environment would agree that corrosion and biofouling are probably two of the greatest challenges we try to manage. So today I'm going to present a concept for designing perforated steel monopiles that will address some of the corrosion issues reported from the inside of the structures, and at the same time create habitat for marine life that addresses sustainability and ecosystem services. And out of interest, the wind farm shown in the first two slides is the Walney extension. They're 87 8-megawatt units, and it's located in the Irish Sea off the Cumbria coast. Well, I've got a lot to get through. I'm going to go very fast. Going to give you a little bit of background of who we are and where we are. Introduce you to monopile supports for those who are not familiar with them. Briefly talk about the corrosion issues. Then look at our knowledge of biofouling and offshore structures. Introduce you to our perforated monopile concept. And then talk about the work that was really done by Monica Mayer, a graduate student here, and a little bit of a summary of what we found. So, as I said, I'm in Florida. I'm a professor of ocean engineering in the Department of Marine Environment, of Ocean Engineering and Marine Science. We're located in a town called Melbourne. And I also run a test center called the Center for Corrosion and Biofouling Control. And our test site basically is at Port Canaveral. And Port Canaveral is a man-made port. It has open access to the Atlantic. And it's an excellent body of water to basically conduct biofouling and corrosion studies. Most of our research is actually funded by the Office of Naval Research, and they actually invest a lot of time and a lot of resources in trying to improve the environmental quality, if you like, of the biofouling control systems that they use. And of course, none of this work is made possible without students and staff, and I'm very fortunate to have a great team. And as I said, the work I'm going to be discussing today is really the result of the research done by Monica Mayer. So monopile supports. These are large steel structures. They're placed in anywhere from sort of shallow to 30-meter water depths, have 80 plus millimeters steel wall thickness, 6 plus meters diameter. They can be over 60 meters long and have over 30 meters driven into the seabed. And as an example, the Walney extension, as I said, has 87 8-megawatt units. And the monopiles have to support a tower, which is 113 meters above sea level. And these 8-megawatt units actually have 160 plus meter rotor diameters. So it's a lot of work is entrusted to the monopile to basically keep these structures upright and functioning offshore. So when you place these structures, the early corrosion control standard basically said the interior of the monopile support would not experience corrosion. And that is because if you seal the interior, the oxygen will be consumed. And once the oxygen is consumed, then basically further corrosion should not occur. However, the challenge of these large structures is actually in creating an airtight seal. And leaks occurred at the conduit penetrations and at the grout seals. And basically that allowed oxygen ingress and corrosion to occur. The initial response to that was actually to place cathodic protection within the inside of the structures. But in a paper by Delweech in 2017, he found that the chemistry within the seawater within these structures caused hydrogen sulfide to form and also created water acidification. And this in itself then created other unique corrosion problems. So the corrosion control update, one of the guidances is that if cathodic protection is used for the internal protection of the inside of monopiles, the the replenishment rate of seawater should be considered, which means now they're accepting the fact that you really need to create some porosity within the structure. So that's a very sort of brief and quick outline of corrosion of the monopile structures. So then we move on to biofouling. And there are thousands of offshore oil and gas structures placed throughout the world's oceans. And probably some of the best studies are off the California coast. And I just put this quote from one of many papers you can find that study these structures. And that's that oil and gas platforms have the highest secondary fish production per unit area seafloor of any marine habitat that has been studied. So they've become actually very productive and important sources of marine life in our oceans. One of the other interesting things with the oil rigs off the California coast was there was a period in the 80s when some enterprising individuals actually got permission from the oil companies to harvest mussels. And so from our perspective, these structures should not just be considered offshore reefs, but maybe potential for aquaculture and other activities. I can go from the Californian coast to the Gulf of Mexico and to the North Sea. And again, just take a random sampling of some of the comments and research that's been made on these structures. But in a 2019 paper by Jennifer, you've got the Gulf of Mexico's hottest diving spots are decommissioned oil rigs. And then I was struck by a comment made by Andrew Want in one of the earlier GloFouling seminars. And he commented that a lot of the structures now are being colonized by Lophelia pertusa. And this is a cold water coral. It grows in deep waters throughout the North Atlantic. And now its presence on many of the North Sea oil and gas installations means that, in fact, should these structures be left in place or should they be removed? And finally, ocean sprawl facilitates dispersal and connectivity of protected species. And in this paper, they make the comment that besides the challenges ocean sprawl presents, novel conservation opportunities such as expanding geographical ranges, increasing secondary production for fisheries, or providing foraging sites for species whose habitats have been degraded. So, biofouling and offshore structures. To us, there can be benefits. Artificial reefs, increased biomass, fish aggregates, aquaculture, habitat heterogeneity, and then this all runs into ecosystem services. You're going to get biomineralization, carbon sequestration, food habitat, and water filtration. Challenges, habitat modification. Well, I think, if you read the papers on ocean sprawl, that is going to happen. And then more from a GloFouling point of view, stepping stone for invasives. And, this is a debate that goes on between the scientists and the ecologists, and it's something that I guess we'll learn more about in the long-term outcomes. But for us, here's an opportunity. Can we combine corrosion control with ecosystem services? And so what I'm going to do is probably do an injustice to the great piece of research that Monica Maher did. And she came up with two hypotheses. The first one was a perforated structure will create an environment with more favorable corrosion mitigation, air quality, and water chemistry. And the second hypothesis is a perforated structure will create a habitat for marine life and recruit a diverse population of organisms. And if you want to read more of her research, she's published it in the August edition of Materials Performance. And the other thing with Monica's research is that she received an Innovation of the Year award from the National Association of Corrosion Engineers. So we've had a lot of fun with this research, and it's obviously received some quite quite a large interest from the corrosion and biofouling community. So experimental design. We're dealing with huge structures. Full scale, maybe a 5-meter diameter, 20-meter water depths. And so what we did is we scaled this down, and we took 1 meter long, 15 centimeter diameter steel pipes. We capped them top and bottom. And in the bottom cap, we put local sediments to imitate the seafloor bottom in the monopiles. And we took 4 of these pipe sections. One was sealed with no cathodic protection to represent the initial offshore monopile design, sealed with cathodic protection, which was the refit to counteract corrosion observed in the structures. And then we put in 2 structures which basically represented our concept of a perforated steel pipe, one with no cathodic protection and one with cathodic protection. And within these pipes, we basically put a small sort of array of test samples. One of the things we needed in the cathodically protected pipes was a zinc anode. And then we put sets of test coupons at the top, center, and the bottom. And we also had the ability to monitor the potential and actually electrochemically interrogate the condition of the steel samples. And just this is a picture of the field deployment, basically of one of our floating test rafts, and the wiring diagram, which I don't really have too much time to go into. And so once a week, Monica would go and measure the dissolved oxygen, the pH, the potential of the pipes, and she would also do what we call potentiodynamic polarization. And at the end of the deployment, we looked at weight loss, and we looked at basically the habitat that was created in the different environments for marine life. So I'm only going to present, a very limited amount of data which she collected. And probably the first thing, because it's oxygen that drives corrosion, was the oxygen concentration. And this is the ambient seawater oxygen concentration. You see it declining between August and September, and that is basically responding to the increase in seawater temperature, at which the saturation levels of oxygen decrease. If we look at the sealed, freely corroding monopile, as you would expect, the oxygen is depleted, but not totally depleted. And this is one of the challenges. To prevent corrosion, you've got to basically remove all the oxygen present in the water. If we look at the sealed pipe, which had the zinc cathodic protection in, again, we saw a depletion of oxygen, but we actually had leakages within our pipes, like in the monopiles, so some oxygen was present. If we look at the perforated freely corroding steel pipe, it basically follows the ambient oxygen concentrations, but both the perforated freely corroding pipe and the cathodic protected pipe, after about a month, they started to show a slight decrease in oxygen. And we assume that was due to the fact that we started to get marine life actually living within the pipe, and their respiration will basically reduce the oxygen concentration. But what about pH? One of the beauties of seawater is that it has a slightly alkaline pH. And, one of the concerns is obviously ocean acidification, but that's a whole other topic. If we look at the sealed freely corroding pipe, we actually get an increase in pH. If we look at the sealed cathodically protected pipe, we get a decrease in pH. If we look at the perforated freely corroding, and we look at the perforated cathodically protected pipes, the pH basically stays pretty constant. And again, I'm not going to get into the detail of the water chemistry, but the water chemistry is pretty well understood. And what was reassuring to us is that the changes in pH that we measured corresponded to the chemistry which we would anticipate occurring in those 2 environments. We then get into potential measurements. And one of the ways that corrosion engineers basically discern whether a steel structure is corroding or is protected is by measuring the potential. And we do this with a reference electrode, which is called a silver-silver chloride reference electrode in this circumstance. We connect it to common of a voltmeter, and we ground the voltmeter to the pipe, and we get a potential measurement. If the potential is more positive than negative 800 millivolts, we're in a regime of corrosion, and that is what we saw with both the sealed and perforated freely corroding steel. And if you're more negative than 800 millivolts, basically you have cathodic protection and corrosion will not occur. So what that allowed us to do is to basically affirm that our systems were working from a corrosion perspective as designed. And again, we can look at the chemistry. And basically, for cathodic protection, I have a zinc anode. The zinc anode corrodes. It provides electrons to the steel cathode. At the steel cathode, you get a reduction in oxygen. You get an increase in pH. And you typically get calcium carbonate and magnesium carbonate. You get chalk formation. One of the interesting things from a biofouling point of view is that increases in pH typically favor the development of benthic organisms that create calcareous shells for their protection. So it should, in theoretic, enhance hard fouling recruitment, enhance growth rates. It forms a stable reef. And one of the other interesting topics which is being pursued by several groups around the world is something called biorock and biomineralization, where people are deliberately placing cathodically protected steel in offshore environments to try and enhance the recovery of things like coral reefs or oyster beds, etc., etc. And again, that's a whole other area of research that is going on. So, at the end of the test, what did we see? Our sealed, freely corroding samples all showed corrosion. They actually had black corrosion products, which is typical sort of iron sulfide and the sort of corrosion that one does not want. In the sealed pipes with cathodic protection, we actually prevented corrosion totally, and the white form there is actually zinc carbonate. So that was kind of interesting. Within the perforated freely corroding samples, we got marine growth, but the marine growth typically was shed as the steel corroded and it exfoliated all the organisms. And the perforated with cathodic protection, which again is our concept, we got pretty well zero corrosion and a very healthy assemblage of marine organisms. If you look inside the steel pipes, these are the sealed pipes. If you look inside the perforated pipes, you can see the contribution of marine growth. And the one on the right with cathodic protection had no corrosion, it had cathodic chalks, and it created a very diverse and stable biofouling coverage in the submerged zone. So what was living in these pipes? And basically, when we removed the pipes from the perforated pipe from the water, we collected everything that was inhabiting in it in a little tray. And there were a few things living in the perforated pipe. But the other thing we noted was because the biofouling was continued to be exfoliated, it actually formed an organic kind of unpleasant mud at the bottom of the pipe. So unprotected perforated pipes, probably not too good an idea. But if we go to the perforated cathodically protected pipes, we found a really rich assemblage of both sort of free-swimming and benthic organisms. And the other interesting thing was the sediments at the bottom of the pipe were very healthy, because they were not being inundated with dead and decaying biology from the sides of the pipe. So what did we learn? Perforated monopile interior walls can be more easily and predictably be protected from corrosion using cathodic protection. Cathodically protected perforated monopiles will enhance the ecology of a region by providing a sheltered space within which marine organisms prosper. And I think the lesson which is probably most important from my point of view is that when possible, ocean structures should be engineered and managed in a way that complements the local ecology and also hopefully provides some ecosystem services. So with that, here's our perforated pipe, hosted with vibrant communities, a little crab and a fish, and we're always delighted to see the sort of amount of marine life that tended to be around this particular structure. And with that, I'll thank you and try and answer any questions that may be presented.